Okay, we are going to finally finish up the chapter on DNA. And this last section has to do with mutations. So when we think of things that can go wrong during replication, transcription, and translation, it's actually pretty amazing that these processes tend to go as well as they do. One of the things that happens is the enzymes DNA polymerase, which does replication, and RNA polymerase, which does transcription, actually have proofreading functions. So if they tend to mess up, they'll actually go back and correct their mistakes most of the time. Of course, they're not perfect, so there is kind of a baseline mutation rate. But some of the things that can go wrong, especially when we think of translation, so let's say that during replication in the DNA, a codon that usually coded for a particular amino acid became a stop codon, okay? And it would tell during transcription, basically transcription would end early, okay? So if that happens and now you have a stop codon on the messenger RNA, the ribosome is going to read that and be like, okay, we're done, when actually that wasn't supposed to happen. So sometimes the ribosome detaches from the messenger RNA too soon. That's going to end up with what we call a truncated, or a protein that's short. It's basically missing a bunch of amino acids. And when that happens, the protein is not going to work very well, if it even works at all. Okay, so that can be a problem. Also, sometimes the ribosome messes up. Okay, normally it's going to select the correct transfer RNA because of the base pairing between the codon and the anticodon, but sometimes it messes up and the base pairing isn't perfect, and so sometimes the wrong amino acid can get inserted into a protein. Now, that could end up being terrible, or it could end up being, we don't really care, okay? Because there are a lot of parts of proteins that aren't that critical to their function. They're not in what we refer to as the active site, or where the key actually fits into the lock, if you can remember back to the basic biochemistry lecture. So it just kind of depends, but it could certainly decrease function as well. So we're going to talk about mutations in this video. So the key with a mutation is the DNA changes, and if it's a mutation, it is permanent. Okay, so if the sequence of bases somehow gets changed in the DNA, that's going to be forever. But okay, the good news, at least in humans, is that the majority of our DNA doesn't contain genes. It's either part of regulatory units, or it's sequences left over from evolution, or maybe, we don't even know what it does yet. Maybe it is a gene, but we haven't discovered it yet. But we kind of luck out because we can build up a lot of mutations that actually don't hurt us because they don't code for a protein. Okay, forensic DNA is a perfect example of that. There are a lot of mutations that build up, but it just allows us to tell people apart. It doesn't mean that a person that has a particular forensic DNA profile is going to be less healthy than a different person. It just, we use it to tell people apart. Okay, and that helps us with, in terms of solving crimes. So I know that the term mutation always has kind of like a science fiction connotation with it, ooh, something mutated and it turned into, something bad. But mutations can actually be good. They can make proteins work better, or they can be bad, or in a lot of cases, we don't really care because it didn't affect either a part of the DNA that codes for a protein, or it doesn't affect a critical part of the protein. But normally when we are talking about mutations in terms of the human genome, we tend to stress the bad part because they usually only become apparent when there is a protein that isn't functioning properly. And, depending on what that protein is, that can result in a really catastrophic genetically based disease. Okay, so sometimes a mutation can cause cancer, and cancer is actually caused usually not by a single mutation, but by mutations building up over a lifetime. That's why we tend to see cancer occurrence in much older people. Not that kids can't get cancer. It's horrible. It's tragic. But if you look at the number of cases, it does tend to be in older adults. Okay. Other times it can make a protein work even better, but that usually doesn't happen to us. It's usually something that happens over, hundreds of millions of years in terms of evolution. So here's some examples of mutation, and I'm actually going to put a short little animation about sickle cell up so you can watch that. Sickle cell anemia is a disease that is the result of a single base pair change. Okay, so think about that. You have 3 billion base pairs, oops, excuse me, and it's a base pair change in one single base pair. It's literally like winning the Powerball, but in a really, really crappy way. And when you see this mutation, the red blood cells develop hemoglobin that is really long and kind of stringy, and it sticks together. And that makes the blood cells, instead of having this nice round disc shape, to have this kind of sickle appearance. They don't carry oxygen very well, so the person is oxygen starved. They also tend to clog capillaries, and that can result in multiple organ failure. And patients basically have to have blood transfusions unless they can get a complete bone marrow transplant to manage the disease. But that is an example, single base pair change, and what we refer to as a point mutation. Okay, so here's some other examples of mutation. Okay, here we have a 6-legged frog, which, I mean, he looks happy. He can probably jump around and catch some flies. The problem with that is that no female frogs are probably going to want to mate with him, because they're going to be like, dude, I'm sorry, I want my kids to have, 4 legs and be normal. And so what we see in nature, because nature is cruel, is when you have these kind of, outliers that are produced, even though they could be potentially healthy, they don't tend to attract mates, and therefore those genes are not carried out in the population. Yeah, nature is cruel. I wish nature was like Disney movies where animals held hands and sang songs together, but unfortunately it's very judgmental and, kind of survival of the fittest. Here's an example of a little lamb, and this was actually a photo that one of my students sent me. She got this from her grandpa, who was a farmer and who raised sheep. So this little guy lived for about 24 hours. And if you look at him, it looks like he has legs and, 4 legs like normal. But if you look at his face, first of all, he has only one eye socket. There's actually 2 eyeballs in there, but, he's kind of like a cyclops. And then what looks like a big fluffy nose is actually his chin turned upside down. Okay, so my point in showing this photo is that if you look at an organism and they're this messed up on the outside, you can bet that they have a lot of problems going on internally as well in terms of their anatomy. Because obviously something went wrong. There was a pretty big mutation, probably a deletion, which means a big chunks of chromosomes were missing. That was early on in a developmental pathway. So it doesn't surprise me, that this little guy didn't live very long, unfortunately. Okay, here's an example of a baby that looks like he has 5 legs, maybe 5, I can't really tell, 5 or 6. But if you look at him from the waist up, totally healthy. Okay, obviously waist down, there's going to be some orthopedic surgery. There's going to be some issues with his limbs, but that had to be a significant mutation that happened later on in development that only affected the lower extremities of the body, because in fact he was completely normal in terms of his organs, and so he's going to live a normal, healthy life. Unfortunately, he's going to have to endure some orthopedic surgeries, but yeah, a pretty big mutation, but it happened later on in the developmental pathway. Oh, here was a 2-faced kitten. Not a 2-face and like, oh, I'm going to lie about you or talk behind your back, but literally had 2 faces. And no one thought this cat was going to live, and it actually lived to be, I think, 14 years old. So, they actually had 2 brains, you know, 2 complete mouths, 2 esophaguses. It was just, everything was joined basically from the stomach on. But yeah, lived a normal life. Okay, here is something called polydacty, and this is also due to genetic mutation. Polydacty means when a person has extra fingers and toes. And so here's an example of a person with no thumb, but 6 actual fingers. Okay, now first of all, I want to define the term inbreeding. So inbreeding tends to have a really kind of creepy incest meaning tied to it. You know, like my dad is also my brother, you know, kind of a hillbilly, you know, thing. Inbreeding just means when you have populations that tend to breed within themselves. Okay, so it doesn't have that, it doesn't mean incest. You know, every population used to be inbred because we didn't have planes to travel places or cars. You know, populations were very isolated geographically. And we still find that populations are isolated in terms of culture, whether it's people from the same religion marrying each other, people within particular racial groups or particular lifestyles. Yeah, so we still see inbreeding, and it doesn't mean that it has to be something, you know, really incestuous or bad. However, we do see populations where there's much more inbreeding. Okay, so for example, this hand is from a person of in the Amish population. And the Amish, and we have Amish that live very nearby, here's in Cedar Rapids, but they are a very isolated population, and they only breed within each other. And unfortunately, it is a very small population. And so you've got a lot of first cousins and, you know, perhaps even closer than that are breeding with each other. And so mutations are not going to get spread out. They're actually going to build up. And so that's why we see things like polydacty and also very, very severe genetic diseases in a much higher incidence in really small isolated populations like the Amish. Okay, it's just due to a buildup of mutations. However, DNA mutations are actually the basis of evolution. Okay, so evolution is, remember, is just genetic change over time, but a lot of times it's based on whatever environment happens to be at the time and whichever traits are most favorable. Okay, so think of this guy, the giraffe. Okay, so giraffes are thought to have evolved from horses, and horses, you know, they look kind of similar, except giraffes have that really long neck. Okay, so, you know, imagine a population of horses and they're all living their best lives and they're breeding. Okay, and then from random genetic mutation, out pops a horse with a super long neck. Okay, and like I said, nature is cruel. So a lot of those, you know, horse chicks are like, ew, dude, I'm sure you're really nice, but you've got this super long neck that's kind of geeky, and I don't want to pass that on to my kids. No offense. Okay, which, you know, you might think that individual would die off all lonely and by itself, but imagine if maybe there was a catastrophic flood, okay, wherever that population of horses was living. Now, all the vegetation that they could graze on the ground is gone. It's destroyed by the flood, and maybe the only suitable food source is up in the trees. Okay, now who's your daddy? Okay, now who's the popular guy that everyone wants to breed with? The giraffe, because in terms of situation and environment, he carries the most attractive traits to be able to reach a food source and be able to survive. Okay, so it really depends on the environment, which traits are selected for, but in terms of the actual mutations, it's just random genetic change. It can be a baseline mutation rate because our cells aren't perfect. Okay, I'm also going to put a little animation about that and how DNA gets damaged just kind of randomly. You know, it could be an environment that's kind of toxic and results in higher, you know, rates of mutation. Okay, for example, laying out in the sun without sunscreen. That can cause what we call thymine dimers, which can lead to mutations. Okay, a thymine dimer is just in your DNA where you happen to have 2 T's right next to each other, okay, two thymines. If two thymines are hit by ultraviolet radiation, they tend to fuse and stick together. And then during replication, they're read as just one T, which completely throws everything off. Okay, that's why that's kind of a scary mutation to have and can give rise eventually to skin cancer. So there are three main types of mutations that I want you to know. Okay, so a point mutation is where one base is simply substituted for another. Okay, and the best example of that I've shown you up till now is sickle cell anemia. Okay, so normally it only affects, because it's only affecting one base, it only affects one codon, and there potentially one amino acid. So it may change the function of the protein if that amino acid happens to be in a really critical part of that protein. Obviously, in the case of sickle cell anemia, it is. Changing that one base and that one amino acid is what causes the hemoglobin to get really long and sticky. Okay, but you can have other mutations that, didn't affect a critical part of the protein, so it doesn't really matter. And we tend to term those types of mutations silent mutations. Okay, what that means is that the mutation happened, yep, it's there, it's a permanent change in the DNA, but it happens to be in a part of the protein that isn't that critical, so it doesn't affect protein function. A silent mutation can also be, it's in a part of the DNA that doesn't code for anything. Okay, so for example, forensic DNA profiles would be examples of silent mutations. Okay, and then we have by far the worst, most damaging type of mutation, which is called a frame shift, because this is where you're going to insert or potentially delete bases, 1 or 2, usually not in multiples of 3, but what it does is it knocks out the entire, what we call the reading frame, and everything downstream from that mutation is going to be screwed up. Okay, so let me try to switch my camera on and show you an example of this. Okay, so I've written a DNA sequence here, and if we remember, for every 3 base pairs equals 1 codon. Okay, so we have codon 1, we have codon 2, we have codon 3, and we have codon 4. Okay, and remember, the number of codons is also equal to the number of amino acids. So here's amino acid 1, amino acid 2, amino acid 3, and amino acid 4. So an example of a point mutation would be, okay, while going through replication, instead of copying the strand properly, and this is a G as it's supposed to be, maybe instead of a G, it inserts an A. Okay, so maybe DNA polymerase makes a mistake. That would be a point mutation, and yeah, it could be critical, but it's only going to change this one codon. Okay, now, if we forget about this and we put this back to a G, an example of a frame shift mutation would be, let's say that DNA polymerase inserts a C right here in between these 2 T's. Okay, now what happens, because remember, every 3 base pairs is a codon, now this is a codon, this is a new codon. So if we call this codon number 1, here's codon number 2, here's codon number 3. You can see they've all completely changed. So probably downstream of this mutation, you're going to end up with all different amino acids. And what you're going to end up with is a protein that likely, if it works at all, works much, much, much less efficiently than it would if that mutation had not happened. So it can be inserting bases, it can be deleting bases, and it basically messes up the whole reading frame in terms of codons and amino acids. So that's why the frame shift mutation is by far the worst. Okay, so let's summarize everything. The bottom line of DNA, Every living thing has DNA, okay, even after it's dead. Its DNA is a pretty hardy molecule, and it hangs around for a while, thank God, as a person who's worked in forensics. DNA is made up of the exact same building blocks in every living thing, okay? So those 4 nucleotides and the 4 bases, A's, G's, C's, and T's. It's just the amount and the sequence of those building blocks that's different and makes different species different. Interesting thing, 99% of DNA is actually the same between people in terms of what genes we have. So, you know, we all have a gene for eye color and hair color, and we can all go to the doctor and have our liver enzymes checked. And that's because we all pretty much have the same genes. We can have different variations of those genes. We call those alleles, but the genes themselves are, yeah, they're pretty much the same. And here's kind of a fun fact. So the only people that have the same forensic DNA are identical siblings, but they're still going to have different fingerprints. Okay, the reason for that is that even if you have an identical twin, you're still laying at different positions in the womb. And actually what forms your fingerprints is waves of amniotic fluid hitting your finger pads. And so your identical twin is going to have completely different fingerprints from you, even though you have the same DNA. So bottom line, if you want to frame your identical twin for murder, you need to frame him with DNA, not with fingerprints, or it's going to come back to you. Okay, so that's actually where I'm going to stop. I know there's a couple more slides, but for the scope of, time, and also what I'm going to ask you about on the quiz and on the exam, this is where we're going to stop. Okay, thank you.